Hello, everyone. My name is Nikhil Sharma, and today I will be presenting my research project in which I realigned clinical studies from the HG38 human reference genome to the telomere-to-telomere genome in order to expose how using older reference genomes may lead to gaps in research conclusions. So, what is a human reference genome? The human reference genome comprises the complete set of DNA within a human cell. Each individual possesses a unique genome, so to compare multiple genomes, a reference genome is used. These comparisons can be used to identify mutations in an individual's genome, which may correspond to genetic disease. The Human Genome Project was the first attempt at fully sequencing the human genome, and it started in 1990 and was declared complete in 2003. The project used the Sanger method of sequencing, and although researchers were successful in sequencing the majority of the human genome, the Sanger method proved too inefficient and slow to fully complete the genome. In 2014, the HG38 human reference genome, which is the most commonly used reference genome, was released. Researchers continue to use it in their studies to this day. However, since 2003, Pacific Biosciences and Oxford Nanopore have developed new technologies that have made sequencing significantly more efficient. Finally, in 2022, the human reference genome was completed using these new technologies in the telomere-to-telomere human reference genome, abbreviated as T2T CHM13. But why is a genome so important to understand? Our genome is the full set of our DNA, which contains code for our genetic information. However, the body does not use DNA to create the proteins that determine our phenotype and genetic traits. Instead, RNA is used to create proteins. RNA transcription is the process of creating mRNA using the DNA as a template. RNA polymerase, as you can see here in this image, is one of the key enzymes associated with this process. Once the mRNA has been created within the nucleus, it leaves to interact with the ribosomes to synthesize proteins in a process called RNA translation. However, before the mRNA undergoes RNA translation, it must fully mature. Upon leaving the nucleus, much of the mRNA is removed and remains in the nucleus. These sections of genetic material are called introns, while the remaining mRNA are the exons. Finally, a poly A tail and a 5' cap are attached to the ends of the mRNA strand to prevent degradation and mark its maturity. RNA sequencing is used to display the gene expression. While DNA contains all genetic information, mRNA represents the genes actually expressed. For RNA sequencing, RNA is converted to DNA as cDNA, and then DNA sequencing is performed on the cDNA. We use RNA sequencing to understand how the genome is expressed. Furthermore, RNA sequencing data is typically aligned to the human reference genome using RNA-specific aligners that consider unaligned regions in DNA represented by the spliced intronic RNA sequences. And for my project, I used the STAR RNA sequence aligner. So continuing, for my project, I downloaded the HG38 and the T2T CHM13 human reference genomes from the UC Santa Cruz genome browser. Then, as mentioned, using the STAR RNA sequence aligner, I generated indices for the two reference genomes. For the project, publicly available renal carcinoma cell line data from the Cancer Cell Line Encyclopedia was used. I used SRA Toolkit Prefetch to download the data from NCBI SRA. Then, prefetch files were split into paired-end FASTQ files using the SRA Toolkit FasterQDump utility with the flag split files. The resulting paired FASTQ files were gzip compressed. Using the STAR RNA sequence aligner, paired FASTQ files were aligned to create a SAM file. And for each alignment, SAM files were converted to BAM format. Then, in Python, each file was iterated through to create a dictionary of counts per chromosome for the read IDs of all primary reads, excluding those with random or alt in the name, to ensure that the reads were a part of the core HG38 genome and not any unplaced scaffolds that were added after the initial release. This filtering was not needed for the T2T CHM13 human reference genome, as there are only core contigs in the reference genome. After sorting the chromosomes, bar plots displaying the number of primary alignments per chromosome were created using the Python package matplotlib, with the chromosomes plotted on the x-axis and the number of primary alignments plotted on the y-axis. Additionally, To ensure that the HG38 and the T2T-CHM13 reference genomes were statistically significantly different, I ran a t-test. My null hypothesis asserted that the number of alignments to both the genomes were the same per chromosome. However, since the p-values, the results of my t-test, for each chromosome was above 0.05, the null hypothesis was rejected. Here you can see I have some of the p-values that I used, but every chromosome had an associated p-value above 0.05. This is one of the bar graphs I made using matplotlib in Python, and this bar graph represents the percentage of reads aligned to the T2T-CHM13 genome per chromosome for the three different samples, as you can see up here in this key. The three different samples are differentiated by their color, so we have dark blue, brown, and light blue, and I use this with my other bar plot for consistency. Um, for the next, likewise, this graph also displays the percentage of reads aligned per chromosome, but this time it was aligned to the HG38 reference genome. Between the two, the percentage of reads aligned per chromosome was higher when aligned to the T2T-CHM13 human reference genome. Additionally, I also found the MapQ score for the reads. Since I used star, a higher median MapQ score means that the primary reads aligned to a smaller number of loci and were more unique. I found that the MapQ score was 100, a high median score. This means that the alignments were broad, but were not broad, but were rather specific to certain locations on the reference genome. As you can see, this is a bar and whisker plot. So, in conclusion, based on my findings, using the updated human reference genome can yield different results in clinical studies that were originally aligned to the HG38 human reference genome. The number of primary reads aligned to the T2T-CHM13 human reference genome is greater than the number aligned to the HG38 human reference genome, indicating that additional alignments may lead to different conclusions or insights. Therefore, I posit that research groups should consider transitioning to the T2T-CHM13 human reference genome when possible, as it may influence results or conclusions in ways that conflict with those derived from using the HG38 genome. For high impact or frequently referenced studies, a shift to the T2T-CHM13 genome is especially important, and future studies should adopt the T2T-CHM13 genome as a standard. And finally, since the current main hurdle with adopting the newer genome is a lack of annotations, research groups should prioritize moving over annotations or re-annotating studies as well. Thank you so much for listening to my presentation. I hope you enjoyed. Thank you.